cancer. Cancer is one of the biggies. And, you know, it's the old thing when you're a speaker, you want audience interaction. You say, hey, guys, how many of you, and you ask people to put up their hands. I'm not going to do that for cancer because for a lot of us, it's just been too painful. It is not something you put your hand up for at a conference. It is one of the great scourges of our time. And we have somebody here who is one of the generals, really, you know, beyond the foot soldiers. He is a general in the war on cancer. He's done absolutely amazing research. As I said, he went from his native Israel. He is now at McGill University in Montreal, one of the great medical research centers of the world. He is a GlaxoSmithKline and James McGill Chair of Pharmacology at that university. His first company that he developed, he is an academic, no doubt about it. He is a researcher, he is an academic. His first company, however, he cut his chops as an entrepreneur, sold it for, I believe, $150 million. Is that correct? In and about that range. He has a new company, HKG Epitherapeutics, and has just done amazing work in the field, and he may, someday his research may save your life. He has come all the way from Montreal to be here. Ladies and gentlemen, please prepare yourselves for Dr. Moshe Szyf from McGill University and HKG Epitherapeutics. Dr. Szyf, so happy to have you. Thank you very much.
We're in the age of DNA, and we believe that by sequencing DNA, and sequencing many, many DNAs, and applying deep learning and algorithms, we'll figure out why some of you will die early and others will die young, why some of you have psychiatric problems and others not. But the truth is, the DNA has delivered much less than we thought it would deliver. And the question is why? And the question ties into understanding how DNA works. So we know for a long time that we have the same DNA in every cell, exactly the same DNA. But one cell develops to an eye, another to a hand, to a liver, to a lung. So obviously, the same sequence can do many, many different things. So how is this possible? What happens with our DNA is that by itself, it's really dead material. So when you sequence DNA, you really get little information because our DNA has to be programmed. And since we're in a high-tech, high-tech environment, let's paraphrase it. DNA is the hardware. The sequence is the operating system. So when you sequence different people, you found about the operating system. But what makes DNA thick is the software. And what it can create is on top of the sequence, which is the operating system, create many, many apps that drive many, many cells and many, many functions in time. So different apps make cells perform specific tasks, and these apps are in what we call epigenetic. They're in how the DNA is marked, is punctuated, in how it's packaged. And actually, on the DNA itself, there is a binary switch. And epigenetics is a binary code for programming DNA. Cytosine, which is one of the 4 letters of DNA, could be either unmethylated or methylated, and that difference can turn genes on and off. And now you can imagine having billion cells with this binary code can program infinite programs. For a long time, we thought that the only thing that writes this program, the coder of this program, is only development. But in the last decade, we have asked the question of what is the role of the environment? For example, we know that the way the mother interacts with the child is extremely critical for the way the child will develop. 2 children with the same DNA can develop very differently based on the relationship they had with their mother. They also will develop differently whether they were born in a rich or a poor place, a place that is highly polluted or not polluted. Many bacteria or no bacteria. Many animals, no animals. How does this work? And for a long time, we had no clue. This was stuff that grandmothers would talk about. Grandmothers would tell you, good mothers are important. But we laughed at them. It's all in the DNA. What difference would it make? If you have a DNA that says that you're smart, you will be smart. If you have a DNA that says you will be silly, you will be silly. It doesn't make a difference what environment you were born into. And I believe that, can you play the video, please? The same way, till I stepped into a bar in Madrid, and I met a colleague of mine from McGill. Montreal is a very, very cold city, and McGill University was built in a way to ensure that there will be no collaboration between scientists. So it spread across the city with silos, and we never meet each other. But when you go to a warm city like Madrid, and you drink a lot of alcohol, you meet a friend who told me, Michael Meany, that this is what he's working on. He's watching how these mother rats are licking their pups, and what happens to these pups later in life. So after a few beers, I thought this is a waste of my hard-earned tax dollars, right? I was a hardcore cancer biologist. I didn't deal with this soft material of motherly loves of rats. But what he told me later was that this makes a difference. So when he watches the animals who were licked highly versus those who were licked lowly, they have difference in stress response. They look different. They behave different. You can go and look at them, and you know which rat had a mother that licked them highly and those that licked them lowly. So I'm a biochemist, and I try to understand what is the mechanism behind this. We spent around 10 years, not before we did this experiment. So the question is, if the DNA is everything, then possibly the mother that licks a lot has a DNA that makes her to lick a lot, and that makes her pups very good, and vice versa. But you can sort out this question. What you do is cross-foster. You split the pups from one biological mother to a different biological mother, and you figure out that it's actually not the genetic mother that defines the phenotype. It's the mother that took care of you that defines the phenotype. So we had the first proof that what grandmothers told us might be true. But how does it work? And we spend many, many years to sort out that there is a conduit, biochemical conduit, between the licking of the mother and the way the DNA is methylated, the way the DNA is programmed. So essentially, the mothers are writing the software on the DNA of their children by their behavior. So that implies that our DNA is not just what we inherited from our parents. It's also what the environment is writing on this DNA. So the coders of this software is not just the history of evolution, but it's also all the things that happen around us. So now, why do I tell you stories about mothers and licking? When you were just promised that I'm going to talk about cancer. So let's go back to the future healthcare horizon. Everybody talks about it. We need to focus on prevention rather than intervention. We can reduce costs of healthcare by early detection. And of course, the challenge is to identify populations at risk for focused intervention and prevention. But the main challenge is everybody's talking about it, but there's very little delivery of it. And the reason is because how do we have tools that can detect disease early, that are accurate, that are cost-effective, high throughput, could be used in a wide geographic area, so no fancy machines that you cannot deliver to a little town. Most people don't live in big cities and don't require highly qualified personnel. So this is the challenge. So when I founded Hong Kong Epitherapeutics here in Hong Kong, I live in Montreal, but my company is in Hong Kong. So I feed a lot Air Canada, taking me back and forth from Montreal to Hong Kong. And there were many reasons why I came here. The first reason was a lot of my research was done in China, and it was the closest place. The second reason was I tried to figure out how to start up a company in China, and I figured out I couldn't figure this out. And the third reason was Invest Hong Kong. A friend of mine that I met in San Diego told me, why don't you go to Hong Kong? So I went and met the leadership of Invest Hong Kong, and I got convinced that this is a good place to actually start business, not to take your business from North America and bring it here, but start it here and then take it to North America. And I think that model will become a more common model. So our goal is to discover early predictions of cancer using proprietary approaches and the immense potential of epigenetics. And then combine it with next-generation sequencing, automation, focus and test for healthy people, and the main point is products should be accurate, robust, cost-effective, and applicable to entire population, rather than boutique, high-end, expensive products. A lot of the genomics research involves a lot of bioinformatics, which involves a lot of money, which could treat very few people and deliver very little impact on the healthcare. So if what I told you is now put it together, our genome is programmed. We have different apps that run our genome. These apps are written during gestation, but also during our exposures and experiences. They keep writing the coding on our DNA. And when a cell becomes a cancer cell, what essentially happened, there's a new app running the cell that tells it to be cancer. And therefore, if we could map this code, we could know who has cancer and who doesn't. So the truth is that genomic technologies have allowed us to determine accurately the level of methylation of predictive DNA methylation cells. DNA methylation is an absolute identity of a cell. It is the software that drives the cell, and if a cell changes its function, you will see it in the DNA methylation profile, and we can map it. But the challenge is to take the big maps and to make them into small units of information that be combined to classify cancer and other diseases. By the way, DNA methylation is applicable not only to cancer, but to any human disease, because any human disease involves a change in the software. So the first place we looked at is the immune system. It's an important participant in progression of cancer. And we discovered, against everybody else who told us that it won't happen, that there is a signature of DNA methylation in white blood cells. So we focused first on hepatocellular carcinoma. And this is what brought me to China, because as you know, in China, hepatocellular carcinoma is an epidemic. 500,000 Chinese are dying every year from hepatocellular carcinoma, and 60 million Chinese are at risk of getting hepatocellular carcinoma because they're infected with hepatitis B. And that brought me to China and eventually to Hong Kong. And in collaboration with one of the largest hospitals in Beijing, we found that there is a signature of DNA methylation in white blood cells. And we took that and developed a small set of markers that together can discover HCC at its earliest stages. So this is where we start. This looks very complicated, but it isn't. So each column here is a different human, a different patient. Each line here is a different gene. Red means it's very methylated, and blue means it's not methylated, and you have anything in between. So as you can see, we could easily classify by these. Here we have 350 sites: control, hepatitis, and liver cancer. And you can see here a sharp boundary as people progress from hepatitis B to liver cancer stage 1. If we can detect people here, we can save their lives probably 100%. If they don't come here, but they come here, they're probably 100% will die from the liver cancer. So finding them here is a question of life and death that can change, not to talk about the impact on the healthcare system that will have to take care of them through these years of suffering. So from all these sites, we went down to 2 sites that could stage cancer very clearly at its early stages, and you can see that they have a very an impressive correlation with the progression of cancer. But there's another way to look at cancer. The immune system is a good place, but also what happens when people get cancer is that they shed DNA from their tumors into the blood, and you have cell-free DNA floating around. And DNA for tumors is found in blood plasma. So we wanted a method that we can robustly and accurately find epigenetic markers in circulating DNA. So there are many, many papers that showed methylation differences in circulating tumor DNA. But if you look at the accuracy, it's very weak. And also the main problem is that they detect other cancers. So the problem is not just to detect having cancer or not. The problem is to know which cancer, right? If you go to the doctor and you do a test and say, oh, you have cancer somewhere, go find the cancer. And then you have to wait till the cancer is big enough so you can see it, and that's too late. So the idea is not just to find cancer, but to find which kind of cancer. So this is an example of a marker that we developed for colorectal cancer and its specificity. So we used around 4,000 individuals that have a DNA methylation map in the human TCGA project. And we mined the data to train the system and then to discover a few sites that could actually differentiate between colorectal cancer and any other cancer. So this is the area under the curve, and we get a sensitivity of around 98%, an area under the curve of 98%. And this is just a comparison between our markers and other markers that are around there. There's only one marker now approved by FDA, which is called Epicolon, which looks at the SEPT9 gene. So we both can diagnose colorectal cancer close to 100%, but the big difference is the Epicolon kit sees many other cancers: breast cancer, cervical cancer, liver cancer, lung cancer, pancreatic cancer, prostate cancer, and stomach cancer. And you can see that in most cancers except stomach, which we see 20%, which is very close to colorectal cancer, we can distinguish very clearly between the colorectal cancer and other cancers. So we're now using this idea to develop markers for liver cancer, lung cancer, colorectal cancer, prostate cancer, pancreatic cancer. The beauty is we have a platform technology, could be easily automated, and we could scan many, many hundreds of samples very easily. I want to move on to other sides of the epigenetic potential. Till now, I showed you data from cancer, but I showed you also data from rats. Are humans affected by environment the same way rats are infected? The first ability, the problem with humans is, you cannot do environmental abuse of humans in a controlled way. So we never know whether people who suffer from abuse suffer because their father had a bad gene and they inherited the bad gene, or was it the abuse that caused the suffering? So God sometimes plays experiments, and in my province of Quebec, there was a terrible ice storm in 1998 that has shut down the entire electric grid of Quebec in the dead of winter, where the temperatures are between -20 and -30. And scientists like me say, we have an experiment now. We have a randomized adversity, and we can now follow mothers who were pregnant during that ice storm and ask the question, what happened to their offspring up to today, which is 20 years later. So we established a score of stress and asked the question, what happened to the DNA methylation of their children when they were 15 years old? And what they did is we correlated the stress to the methylation. Now you know how we do this. Every column here is a different child. Every color is a different gene. And as you can see, as stress increases, these genes become more methylated, and these genes become less methylated. So the ice storm is written on the DNA of the children. And then we ask ourselves, can we establish now a score, like we established for cancer, for the stress? A methylation score with just a few genes. We could establish a very good methylation score, which we believe will be a very powerful tool in the hand of psychiatrists who are trying now to have some objective measures of early life adversity. So can we now think about DNA methylation biomarkers as actually tools for a dynamic, lifelong, personalized management of health and health risks? Because if on our DNA, our life story is being written, it's a dynamic story that we need to follow. And can we combine DNA methylation markers with dedicated interventions? So you come to me and I test your methylation, and not only I tell you you have a problem, but I will provide you a solution and then a continuous assessment of progress using DNA methylation markers. So one of the fascinating things about epigenetics is epi-aging. So we founded a company in the park with another company called Govita and other partners to harness the power of the epigenome for healthy aging. So we have a clock in our DNA, which is a methylation clock. And this clock predicts our aging. It's affected by our life history. And advancement of the clock means we'll die younger. And if the clock ticks slow, it means we'll die older. And there's many studies already that have established that this DNA methylation clock is the best predictor of your death from any cause at early age. So if we can perform personalized intervention to slow down the methylation clock, we can slow down biological aging. So we took that clock and brought it down to our assay, to our multiplex next generation assay, which is simple. And our idea is that our assay should cost between $10 to $20, not $1,000 or $5,000 or $10,000 like they cost today. And here you can see how this clock predicts the biological age and the real age and the DNA methylation age. But what you can see here is some people already there are around 800 people here, and you see some people are aging much faster than the real age. Their DNA methylation age is much older than their real chronological age. So we would like to have this assay done, hopefully on the entire population, by which the way you take your HDL and LDL and blood pressure, you can measure your DNA methylation age and intervene, and then see if you have moved your clock backwards and forwards, and apply to it some sort of a learning that we can keep learning from the results of your study to give you improved solutions. So our idea is that we can use nutrition and lifestyle to move your clock backwards and to give you more years of life. And we will do it in a very smart and learning way by which we keep learning from the changes that we have caused to your DNA. How good were we with the intervention? So we're not going to give you interventions just because it looks cool to do this, but because we can see by a real read in your DNA that intervention is working. And I believe the future of healthcare is in this, is in managing your life by looking at the software that programs your life continuously and dynamically and integrating it with appropriate information. So a long-term goal is to have a lifelong dynamic interplay between DNA methylation assessments and health-prone interventions, and to develop a lifelong roadmap for preventing disease and increase well-being. And I think that kind of technology will change the way medicine is working because it's based on a thorough understanding of how our DNA works and how it interacts with the world. So when we started with DNA, we called it the book of life. But DNA is really the movie of life. And it is an interactive movie. You're holding the remote control, and you're removing and adding actors as you go through life. And this is the big difference between genomics and epigenomics. Between looking at DNA as a book or looking at as an interactive movie. And we have now the tools to provide people with guidance on how to keep moving their movie in a way that improves the narrative and the end of that movie. We also learned that that movie not only includes our own lives, but it includes experiences of past life. The challenge of technology is to take this, make it cheap, make it robust, make it accurate, and make it applicable not to a few rich people, but to the general population. Thank you very much.
Wow. Thank you so much, Dr. Szyf. Absolutely brilliant. I think we might have time for a couple of questions. We got 5 minutes for questions. Dr. Szyf, how much you want to say about the business, HKG Epitherapeutics? Because you have this strange situation where you're out of Montreal, you're doing partnering with people in China, and then you've established a company here in Hong Kong. Can you just comment on that a little bit, and some of the thinking behind that?
So the idea is that to move these things faster, you have to start them in Asia rather than in North America.
Wow. Okay.
And to do the things that I already have done here in the first year, to do it in North America, I would have needed a 10 times investment.
Right.
Because doing business in North America is extremely expensive. And most of the expense has nothing to do with the research.
Right.
It has to do with all the paraphernalia that you have to put around it. And the second thing is that Hong Kong is unusually friendly for business.
We like to think so.
And it, having worked in many, many different countries, I think it's a very nurturing environment for business. Mm-hmm. And my team is an amazing team.
Okay.
And that's the third element. You can find here really good people. So my idea is not to go first to the FDA and go to the CFDA.
Right.
My idea is first start here. Develop a powerful product, and then when it's so powerful that even the FDA will succumb to its attraction, bring it to North America. I think it was the best decision I've made.
Okay.
This is the best team I've ever had.
Right.
And I've run a laboratory in Montreal, as you know, for almost 40 years.
Yep.
And
you still have the team in Montreal.
Of course.
And they have a role to play.
They are good, but I think the team that I have here is really excellent.
Okay, and that's another team in Hong Kong.
Yes.
And then research partners in China, so it's really the triangle.
Right.
Okay, sounds good. We got some questions coming in. Some people want to get right into the science. Let's see, how specific is the methylation level to cancer detection? Do you think, which one of those do you want to tackle first?
I will start from the first. So how specific I showed you. There's definitely no reason why you shouldn't approach 95%. No reason.
Yeah.
And there's no reason why you shouldn't approach almost 95% the specific cancer, no cancer. Mm-hmm. When I look at the public data, most of what we didn't cost us any money. It cost us some brains. And because the data is there. I think our problem in the world is not data. It's trying to look at the data. And when you look at the data, it's so remarkable how exquisite those patterns are. Mm-hmm. The challenge is how to make it into a product. So this is what we're working on. There's a difference between science and a product. And this is what we're working on. So
okay, new questions coming in.
How can it,
yeah?
Yes, I think it is extremely important for insurance to partner with us because we can reduce, if not 95, if 50% of the HCC cases would be detected, for example, colorectal cancer would be detected at a stage where you go to the doctor for half an hour operation and it's done, not only you save the life of the person and all his relatives, and we know how cancer hits not just the person, but the entire
whole family.
environment,
Yeah.
but also the economy, because this guy will come back to work rather than sit in hospitals, and the health insurance. So I think they should partner also this lifelong follow-up and force their insurers to do it, because if we can have a way by which can assess health and we can improve health, of course you can do a simple economic calculation, what impact does it have? Even 10% of reduction in morbidity.
Catch it early, a fraction of the cost. I want to make sure we get this one before it goes off the screen. If we have piles coming in, early detection of cancer, false positives. Can you get false positives and unnecessary tests, or is this more
So we
reliable?
design our test that we will have no false positives. So that's one requirement.
Okay.
And so we have many restrictions on the test, that because we have so much data on how normal cells look like, it is possible to do that.
Okay, there's one that's going to disappear. Changes to DNA are passed on transgenerationally.
That's the most exciting aspect of it.
Yeah.
It's a very active field. We and others have been working on it. I think it is. We don't know what fraction of it is, but there is definitely strong evidence that it passes.
So the nurture can get into the nature.
Of course.
Of course.
Of course. It's simple chemistry. It's simple coding.
Yeah.
It just writes the code on our DNA.
The software. Which
The software.
Tech guys get. Let's see. What challenges do you see to make Epic CRC a screening standard and clinical protocol? Wow, we're really getting technical here. Is that something you want to tackle, or is that a specific?
I mean, the challenge here. That goes very fast.
Yeah.
Yeah. So I mean, the challenges here is to essentially basic marketing challenges.
Yeah.
Is convince the doctors, convince the patients that that's good for them. And I think the way to convince them is by producing better and better data.
Right.
And that's the challenge.
All right. We got time for maybe one more, autophagy or timeline for commercial. Let's go timeline of commercialization, because I know we got a lot of the investors here today.
The beauty of diagnostics is the timeline for commercialization is very, very short. And our technology is so powerful that we can very fast design an assay that could be commercialized. And as you heard in Hong Kong and many other countries, there are no restrictions on regulations of tests. And therefore, nothing stops us even today from going to the private market and asking people, you know what, we can offer you this test. You don't have to rely on it 100%, but it can offer you some suggestions. So I think the time to marketing is very, very rapid. Of course, finally, marketing into health insurance and to governments will take probably longer.
All right. Well, Dr. Szyf, you've done an absolutely spectacular job. You're a researcher, but you're also a businessman, so we know that you're on the right track. Ladies and gentlemen, Dr. Moshe Szyf. Thank you. Thank you. Thank you.